To request services from the University of Arizona Genetics Core, please make sure you're logged into the iLab operations software and at our core's landing page. From here, click Request Services. This will pull up a list of available service types. To request low-volume sequencing, click here to expand this category, and over on the right, click Request Service. This will open up the low-volume sequencing service request form. Please be sure to complete all required fields indicated with a red star. This sample grid right here will allow you to inform us your sample names, the primer or primers to be used, the concentration and length of your template. Let's fill these fields out here. Sample names can be typed in one at a time, or you can paste. This field will be where you tell us which primer to use on the sample. You can choose from the drop-down list containing our house primers and drag to populate down, or alternately, if you have your own primer, you can enter that here. If you choose to use your own primer instead of our house primers, please be certain to send the primers along with your submission. It is not necessary to use more than one primer to sequence each sample, but many people find this helpful. For example, you may be conducting a forward and a reverse sequencing reaction on these samples. If you are sequencing each sample with more than one primer, be sure to indicate separate primers in separate columns. Please tell us the concentration of your sample in nanograms per microliter. It is not necessary to give us the units, just simply the concentration. And the template length. This is a 600 base pair product. Again, you can drag down to populate. If you would find it easier, you can download this plain grid into Excel, fill it out, and then upload it back to here. Next, you're going to indicate the total number of reactions. Please be sure to count each primer and sample pair as a reaction. In this example, I have 10 samples, but I'm using a forward and a reverse primer on each one. So the total number of reactions is 20. Enter the number here and click Add Selected Services. Next, you'll indicate the template type. Often they're PCR products, but we can also perform sequencing on the following template types here. Please keep in mind this may affect the amount of template required, and please see our FAQs to ensure that you're providing sufficient template or input. These are PCR products. These settings don't apply to most situations using DMSO, max volume, or a two-step PCR. If you don't commonly select these options from our lab, please leave them unchecked here. If you have questions about whether or not these may apply, please contact our laboratory prior to completing your submission. Finally, we'll ask you to read and acknowledge the end of project terms. You'll have to acknowledge this statement each time you submit a job to our core. Once you've completed all required fields, click Save Completed Form. Based upon the number of samples and the cost per sample, this has generated a total projected cost. If you are a U of A internal user and you have access to U of A funds, you'll be able to select your account number here. If you're an external user, or you do not have access to U of A funds, please enter a PO number here or type check or credit card to indicate your payment type. Feel free to click on the View Form link to review your submission. Please be sure that all the samples and indicated primers are correct prior to submitting to our core. When you're satisfied with this form, click Submit Request to Core. Now we're back at our dashboard. This is the submission we just created, our low-volume sequencing submission. It is very important that you locate the numbers at the end of your service ID and copy and transcribe these numbers onto your physical submission. For low-volume sequencing, you're submitting individual tubes, so it may be easiest to use a label printer or to simply attach this number to the container that your samples arrive in. This might be a box, a plastic baggie, or other container. This number will be critical for our laboratory to properly identify your shipment when it arrives. If you have questions about how to use iLab operations software to submit for low-volume sequencing or a different service request type, please contact our laboratory by emailing us at uagc@email.arizona.edu. We look forward to working with you.